protein structure and synthesis. And to start with, we're going to look at protein structure. Proteins are made of chains of amino acids, and there are 20 different amino acids to choose from. Amino acids are made of a part which is the same for all amino acids, it is identical, and a side chain with different chemical properties depending on the amino acid. So how do we get from amino acid to proteins? Now, proteins are made of chains of amino acids, and these amino acids are bound together by strong covalent bonds called peptide bonds. This is what a protein would look like with between 350 and 400 amino acids. Here the detail of each amino acid is not shown, just the overall structure and shape. Indeed, proteins are long chains of amino acids which then fold and twist with bits that may be added and bits that may be removed. All that needs to happen before the protein can be considered finished and functional, i.e. able to do its job. So how do we go from a gene, i.e. a piece of DNA encoding for a protein, to the protein itself? This is what we're going to see next, and it's called protein synthesis. We start with the DNA, and the DNA is found in the nucleus. Now, you've learned at the start of the course that protein synthesis was taking place at the ribosomes. And as you can see here, these ribosomes are found in the cytoplasm. So, there's a technical issue because the DNA cannot get out of the nucleus. It's just too bulky. There are some holes in the nuclear membrane, but these holes are too small for DNA to pass through. So what we need is to make a copy of the genetic code, i.e. the order of the bases, and this copy should be small enough to squeeze out of the nucleus. This is shown here in red, and as you can see, the copy can get out of the nucleus and carry the message from the nucleus to the ribosomes in the cytoplasm. This messenger is called messenger RNA, abbreviated mRNA, and it is made of a slightly different nucleic acid to DNA, but very importantly, it is short and single-stranded, so it can maneuver easily through the holes of the nuclear membrane. Let's now look more closely at the first phase of protein synthesis. The enzyme that assembles mRNA first unzips the DNA. Once this is done and the code is accessible, it assembles the RNA using the DNA strand as a template. Finally, when the mRNA has been synthesized, it leaves the nucleus to get into the cytoplasm. Before we move on to the next part, let's just spend a bit more time on mRNA and summarizing what we've learned. mRNA stands for messenger ribonucleic acid. It is a nucleic acid like DNA. It is single-stranded. mRNA can be found both in the nucleus and the cytoplasm, whereas DNA is only found in the nucleus. And finally, mRNA contains the base uracil instead of thymine. This is an example of the fragment of mRNA, and you can see that there are no thymines there. They've all been replaced by the base uracil. Now, this base uracil, the same as thymine, is complementary to adenine. Now, although you don't need to know about uracil for N5, you might come across it by watching other videos, so you might as well be aware of it. So, we now have understood how a copy of the genetic code can move from the nucleus to the cytoplasm. It is therefore time to explain what happens afterwards. The second part of protein synthesis takes place at the ribosome. The ribosomes assemble at the start of mRNA. The amino acids are present in the cytoplasm. They're attached to a carrier molecule, and each carrier is specific to an amino acid. The carrier contains a triplet of bases, shown here. If the triplet of bases of the carrier is complementary to the bases of the mRNA, then the carrier with this amino acid enters the ribosome. Its amino acid is now ready to be attached to another amino acid. The ribosome moves along, and the new amino acid, corresponding to the next triplet of bases, is placed by the carrier next to the first one. A covalent bond is formed between the two amino acids, and the ribosome moves along. The process repeats itself, attaching amino acid one at a time, following the genetic code, i.e. the sequence of bases on the mRNA determining the sequence of amino acids in the protein. To finish this video, here is a summary of protein synthesis and protein structure. Proteins are made out of chains of amino acids. There are 20 different amino acids. Messenger RNA is another type of nucleic acid. It is single-stranded. It can be found both in the nucleus and the cytoplasm. The base sequence from DNA determines the amino sequence in a protein, and the messenger RNA carries a complementary copy of the code from the DNA in the nucleus to a ribosome in the cytoplasm, where the protein is assembled from amino acid. Thank you.